Bonjour. I'm sorry to say that my presentation will be in English. I will try to speak very slowly for those who are not fluent in English, and please, you feel free to ask me questions in French, because I understand French. So what I would like to share with you today is a little story about hybrid hydrogels. And in particular, I would like to show you why hydrogels, what can we do with them. And of course, I would like to introduce you why we are interested in hybrid hydrogels. So that means a composition between a soft material that will be essentially a cross-linked system and nanoparticles, in this case, mesoporous silica nanoparticles. And I would like to show you what can we do with these materials. So I will introduce the first experiment in vivo. So we use this hydrogel as materials for implants, but also to fill fistulas and problems related to intestine and stomach disease. So I would like to share with you the first results on this area. And of course, as any material that you are going to use in vivo, this material must be perfectly biocompatible, eventually even biodegradable for some applications. And as you might know, most of these silica nanoparticles that we're going to use, these mesoporous silica nanoparticles, are quite large, between 60 and 100 nanometer, and they have a problem that they are not so biodegradable. So I would like to share with you a way that we can make this particle degradable, and indeed they are not degradable, but can be breakable under a certain stimulus, and therefore they can be reduced in pieces smaller than 5 nanometer that can be excreted quantitatively from the body of the animals. So this will be a little bit the menu of this morning talk. And let me start with a small introduction for those who are not familiar with hydrogels. So these are often cross-linked materials that could be cross-linked by covalent linkers, or because you use non-covalent, typically physical interactions between units. Maybe some of you know that some of these hydrogels are already commercially available. So you indeed eat some of them when you eat these jelly candies. Or some of you maybe have used this silicone hydrogel for contact lens. These are the contact lenses that you use only for 24 hours and then you displace, you throw them away. And there is already some example of hybrid hydrogel where, for instance, Aida in Nature 2010 shows that he can use some kind of platelet. These are clay material, where these were introduced in a system that could be cross-linked physically in this case, to obtain self-healing hydrogels. So our approach was very simple. So we use a reaction that can be conducted at room temperature. And this is essentially a Michael addition reaction that can involve a different type of amines and different type of these keto derivatives. And what is interesting in this case is that we cross-link the material, so we attain a material that is a polymeric structure that in the collapsed form, as you can see here, has a very, very small volume. However, when you start to add water, the volume can increase more than 1,000 times, because this material is able to swallow the water, so to entrap the water into the net that are formed upon this reaction, and therefore you can have a material that swallows dramatically. So you can do this with FDA-approved components or with biocomponents, if you like it. So we can use, for instance, this type of amines. These are spermines. You can use a gamma-aminobutyric acid that is also a biological component. And when you cross-link this material, you can obtain gels that have different, as you see, appended groups. So we can functionalize with many different systems. Eventually, they can have also carboxylate, free acid. And this material has a very nice elasticity. They have a self-healing properties for the first few hours. They have a reversible swelling. That means we can dry the material, we can re-swell the material. In this case, this particular hydrogel contains 86% of water. If we look what happens when we expose this material to cells, you can see that the cell, in this case, these are HeLa cells, can indeed grow on the surface of the material. They can proliferate, eventually can also indeed penetrate into the gel. So if we modify, of course, any of these components, you can have different type of hydrogels, and this means different in the way they form this net structure, and so therefore you can modulate the system. However, one of the problems with these gels is that you cannot really fill the cells since you cannot deliver any nutrients to the cell or eventually any growth factor if you are working with a stem cell. And so we thought that this could be accomplished by combining mesoporous material with these gels, and in particular mesoporous silica. So for those who are not familiar with mesoporous silica, these are nanoporous materials that can be made by template synthesis. So typically you use a micelles or a polymer that is used to template the formation of silica. And what is nice about this synthesis is that you can tune the pore size, but also, of course, the material size and the aspect ratio of the material. So if you empty the pores, in this case, we use, you see, nanoparticle of about 50, 60 nanometers, where we can have a possibility to fill these pores with different molecules. And in our case, I will show you in a moment, we can use nutrients for the cell, but we can also use other type of factors, for instance, for stem cell differentiation. And so the idea is now that this nanoparticle can become one of your nodes for the cross-linking system. So in other words, if you take your nanocontainers, your porous silica, and you functionalize your system with amino groups, these amino groups can act as a cross-linking agent, exactly like you saw for our spermine system. And so when you do that, of course, into the net of your hydrogel, we will end up to have some of these nanocontainers forming the structures. And for us, it's enough to have just few percent, even 1% of these materials, in order to release components into the gel structure. So what is interesting, of course, is the nanoparticle can be seen because you can put fluorescent dyes. But what was very surprising for us is that the swelling behavior of this hydrogel is very much improved by the presence of these nanoparticles. In particular, we have a much more reversible swelling and collapsing behavior. We have a much better elasticity of the system, and we have more water that is entrapped into the gel structure, up to 95%, depending on the gel composition. What is also interesting is that the material is self-filling. I will show you an example. That means you can cut the gel and reattach without noticing where it has been fractured. So, of course, the first question was, well, is this gel also biocompatible? Will the cell indeed proliferate and grow in this system? And what you see here is a bright field visualization of the cell inside this material. And you can stain the cell nucleus, typically with DAPI or with ERST. And you see very nicely that the gels do not only live and proliferate, but also tends to fill the entire space of the gel. So they migrate through the entire height of the gels. This is just a 3D view of the gel, and this is a top view. And so at this point, of course, the next question is, can we really release some kind of factors to help the cell to proliferate and to live? And so what we have done was essentially to repeat the same synthesis, and we have just loaded our pores with a different type of amino acid in order to favor this cell proliferation. And when you do that, of course, you can repeat the same experiment. And now you see that the gel, as was before, of course, is perfectly transparent. It has pores that have a size, depending on the type of amines we use, between 30 and 100 micrometer. You see one of these example here. And again, the gel presented this very nice self-filling ability. So how can we grow the cell in this type of hybrid hydrogel? Well, it's very easy, because indeed we can mix already the cell culture medium containing the cell that you want to grow inside your pre-gel mixture. And when you do that, essentially you obtain again a solution that will be jellified, depending on the type of cross-linking system, between 20 minutes and 2 hours. And again, what is interesting here is not only the time of jellification that is quite short, but also the fact that we don't have to add any other chemicals, and we do not have to heat up the system. So this is a room temperature formation of this gel. Of course, you can analyze again the gel in this case, and what you notice is that if you look at the host localization, so this is again label for the nucleus of these cells, you see very nice that the cells are nicely distributed all over the places. And for us, what was very nice is after 48 hours, we have almost no mortalities of the cell. And this was very important because, of course, now if you want to imagine to use this material, for instance, for tissue regeneration, you wanted your cell grow and proliferate for more than a week, because eventually this is the time that the stem cell needs to differentiate into the system. So you can see indeed that the mortality of the cell using a way to detect the dead cell, and therefore we can have a good overview of the stability and also of the viability of the cells in this media. So at this point, of course, we were wondering what is the time of the release of these eventually nutrients from this mesoporous material. And so we did the opposite experiment. So we did not label the cells, but we put the labels inside the mesoporous material in order to see the release of the labels, because eventually this label would have colored the nucleus of the cell. And so this is the same experiment, and now you see very nicely that the UXT is uptaken by the cell as you would expect, and it would color the nucleus of the cell. And so therefore you can monitor the release of this component from the mesoporous material. So we were quite happy, and at this point we were wondering what is the real role of this mesoporous material and the release of these nutrients. And so we did an experiment over 7 days where we followed the growth and the proliferation of mesenchymal stem cells. And what you see here is the comparison between the hydrogel containing, in this case, the mesoporous material that have been filled with a growth factor. I will show you in a moment the formula. And the same material without the nanoparticles, so this is just a pure hydrogel. And you see very nicely, and I don't think I have to explain too much, that in the case of the presence of the mesoporous material that can release this growth factor, the number of cells that populate the gels after 7 days is much, much higher than the normal hydrogel in the absence of these mesoporous systems. You can also follow this by fluorescence spectroscopy, and you see very nicely again that the entire thickness of the gel is populated by the cell, even though these are very thick gel, as I showed you in the previous picture. So this was very encouraging, and one thing that I mentioned to you at the beginning was also that we can self-heal this gel. So in other words, if you cut the gel in pieces and you color some of these pieces, for instance with methylene blue, you can reattach the gels and they form a unique structure where you can barely recognize where you have cut the gels. And this was a very interesting properties for us, because we were indeed wondering what happens in the moment we will have the reservoir of the food or this growth factor only in one part of the gels, to see indeed if the cell can migrate towards the gel that contain indeed the growth factor. And so what we did in this case was to fill the mesoporous material with this stromal cell-derived factor. This is a protein that is called just shortly SDF1 alpha. And the idea was that, of course, if this can be released, eventually the stem cell should migrate towards the section of the gel that contain this material. And so the idea was, of course, to do exactly what I showed you before, to fill this material inside the mesoporous system, to create the gel, and then to see what happens. So again, of course, this forms obviously normal gels because the material is inside the pores. And in order to understand better, indeed, if this release of this growth factor could have a chemotaxis effect on the cells, we build up a very simple device where we have essentially two cylindric system. The first cylinder is this inner core that you see here in blue, where we have essentially formed a gel containing our nanoparticle with a growth factor, and we have colored this with methylene blue in order to see it. And then on the external cylindric part, we have just have added a gel containing the cells, and because of the self-filling factors, of course, at the end you have just this cylindric blue zone and the white transparent zone that are formed by the same gel, with the difference that in the center we have the nanoparticles that contain the growth factor. And what you see indeed is that you can barely distinguish where is the fracture, and the idea was to see if the gels that are contained only, the cells that are contained only in the gels, that is in the outside ring, could eventually migrate towards the blue ring because they have the feed factor. And what you see very nicely is indeed that you can really observe in real time the migration of the cell. They have a very interesting trajectory. This is now understudied by the biophysicist. And what you see very nicely is that already after 7 days, almost all the cells have reached indeed the area where there was the release of the growth factor. And if you want to see this really by microscopy, you can see very nicely staining the cells that there is a definite migration of the cell towards the inner core of the system. What is interesting is the cell migrate, so they take about 3 days to reach the area where there is this gradient release. And they do this in a very nice way. They do this in a linear trajectory mode that is very unusual for cell motion. So understood that, of course, we realized that perhaps this material could be used as an implant because this cell eventually can differentiate in different tissue. And therefore, what we have done was to form this gel, including this mesenchymal stem cell, and we have introduced it in a pocket that the surgeon have created between the muscle and the bone of these little mice. And after 1 week, we have reopened this wound, and we have looked where the gel migrated eventually. And what you see is that the gel stayed perfectly on place where we have left 1 week before. And what was interesting is that there was no sign of inflammation. And in order to study this immunogenic response, we really looked at the different gel, that one without the mesoporous material that can release indeed this growth factor, and the other one after 1 week where we have indeed the growth factor. And by comparison, you can see very nicely that in the absence of this growth factor, you form a scarf. So you have an immunogenic reaction. And you see indeed that the cell colonize and organize along this line here, while in this case, you don't see this formation of these fibrotic areas of this immunogenic area, showing indeed that the material has been perfectly integrated into the tissue where it was implanted. So this is a very exciting result, and at this point we thought, okay, maybe then we can go to have this in vivo in a different way, because we thought that perhaps this material, since it is a liquid, as you saw, and becomes a solid in 10 or 20 minutes, could be used to fill the fistula or the ulcera of the intestine in bigger animals like pigs. And so the idea was to create, of course, artificially in ex vivo transplant at the moment. Now we have already in vivo, indeed, some of this damage in the intestine and in the stomach of the animals, and to see if our material could fill these holes, these tubes, and eventually could be perfectly biocompatible. So the first question was, can our gel survive into the stomach, where, of course, the pH is very dramatic, it's around 3 or even lower in some places, and of course it has to resist for many, many days. So the first things we have done was to modify slightly the structure of the gel, and we have looked at different gels containing different type of amino groups in gastric juice. So we left the gels, and you can see now the gels here that have been colored for more than 7 days. Now we have it indeed for 3 months into the gastric juice, and then we were just taking a little piece to see the decomposition eventually of the gel. And what we noticed was that our cross-linked material was very resistant in this condition. So once formed, essentially can really resist in this dramatic situation. And also what I mentioned to you is that the gel is a liquid, so can it be easily transported wherever we want through an endoscope or through a tube into the stomach. And therefore the beauty was that indeed it could be injected as a liquid and then eventually solidify. And so this is, I'm sorry for these 2 pictures, but I want just to show the real experiment where we have colored again the gel with methylene blue just to see it. And we have injected in a part of the stomach where there was a fistula that was artificially created. And what you see is not only that this gel is filling the fistula, but when you cut the material only after 20 minutes, the gel has been completely formed and has filled all the space in the mucosa of the stomach that was necessary to fill. So this was, of course, very exciting because it means that the material is not only adhesive to the mucus, that is a very unusual properties, but also it stays in place and is solidified in a very short time. That is what we want, because we don't want the surgeon waiting for hours with the stomach of the patient open. So this is where we stand, and of course we are going on with this material. But the next question is what happened to the nanoparticles? Because once, of course, that the nanoparticle have fulfilled the duty of releasing eventually drugs or growth factor, we have eventually the problem that this nanoparticle will be accumulating into the organs, and therefore they will stay wherever we left them into the gel. And the other question was, we can deliver with this mesoporous particle, very small molecule, but what about if we want to deliver larger molecule like enzyme or larger proteins? Can we devise something in this respect? And so what we thought was to realize a system where our particles are not only made of silica, but they are made of organosilica. And so in particular, we devise some kind of capsules that are able to wrap around a large molecule like around enzyme or proteins. And they are made indeed of an alternation of silica groups and then what we call breakable groups. So these are just small molecules that can break under a certain stimulus. That could be, for instance, pH, or could be a redox reaction, like in the case of the disulfide system, that upon addition of a reducing agent can break entire groups. And so when you do that, of course, you will break this blue box, and therefore the material, like an egg, will break in million pieces. So this was the idea, and the idea was, of course, are the proteins and the enzyme, once they are entrapped, able to be still active, and so therefore to perform the biological function. And so this was, this is the cover of the Young Evante. So the idea would be that the capsule will stay intact until they reach eventually their target, or they are indeed inserted in the gel and into the stomach. And then they would break only when they start to interact with cell or even being internalized into the cell. So this is the results of the encapsulation of a cytochrome C protein. So we just use this small protein because we can easily visualize the encapsulation looking at the IME group of the cytochrome C, that is this porphyrin. And what you see is the formation of these capsules that are very nice. They are very monodispersed. They are about 40 nanometer in size. And what we were expecting indeed to see is that this capsule can eventually fragment in very little pieces containing this thiol group upon addition of the glutathione. We use an 8 millimolar solution of glutathione that essentially mimic the same concentration of glutathione into the cells. What is also interesting is that, of course, in the moment you break the capsules, the protein will be released, and so you can just monitor the release of the protein into the supernatant in water, and therefore you can compare the non-breakable capsules, so the normal silica material, with this breakable system. You see very clearly that the protein is released, and upon spectroscopy you can measure the amount of proteins that is released, that of course correspond to the amount of proteins you have been able to encapsulate in these 40 nanometer particles. In this case, we have been able to encapsulate 34 proteins per particles, as you see here from the increase of the kinetics of the release of the protein into the water system. Also, what you notice is that the breakage can be controlled, and in our case can be released already after 1 or 2 hours of encapsulation, depending on the amount of numbers of the groups that form the silica structures. So just to conclude, I want just to show you a couple of more pictures, because indeed people wonder what happened to this uptake of the cell using these specific capsules. Are these still able to be internalized by the cell? And the answer is yes. With our great surprise, the internalization of this breakable capsule is much, much faster than pure silica. They are encapsulated in 7 minutes. And as you can see from the green fluorescent protein that has been encapsulated, the particles are in the cytoplasm. And you see very nicely that indeed using green fluorescent protein, you can monitor not only the internalization, but also the release of the green fluorescent proteins that spread all over the cytoplasm after a certain time. So if you don't believe confocal microscopy picture, you, I think, will be convinced if we look at the TM inside the cell of the non-breakable particle versus the breakable material. And you see very nicely here that the non-breakable material, after 48 hours, they are still intact. You can really recognize the particles. While in our case, the material not only is broken, but is excreted by the cell into the cell culture media. So this is very exciting, and as a last slide, I would like to show you that, of course, this is true not only for this type of proteins, but it's also true for proteins that can be active towards, for instance, apoptosis, so to kill cancer cell. And so we have tried, together with Don Hilbert from ETH in Zurich, a protein that is synthesized, it's called onconase. This is a protein that selectively will kill glioma cells. And we have tried to see indeed if our capsules were able to release this protein, if the protein could still kill this glioma cell. And you see here the viability after only 24 hours, and nicely you see that the protein is still active, and therefore it can express its killing activity. So this is, I would like at this point to conclude, and I would like to leave the conclusion to you because I would like to have a couple of minutes for questions. And therefore, I would like just to thank the people who did the work with me. In particular, all the breakable material have been developed by a single postdoc, Dr. Prasetianto, who is a fantastic person in my group. And all the work on the system, also in terms of biology, was studied by Dr. Gabrera. And most of the hydrogel work is done by only one person, that is Federica Fiorini, and now together with Mike Tettinger. So I would like to thank them. I would like to thank the hospitals who help us with all the in vivo research, and in particular the Methodist Hospital in Houston and our institute, the hospital in Strasbourg, together with the IRCA, the Institute of Research for Cancer of the Digestive Apparatus. And of course, I would like to thank our sponsors, and thank you very much for your attention.